All right. Good morning, everyone. Welcome to our weekly Wednesday morning orthopedic surgery grand rounds. So today we have a special guest speaker, Dr. Adam Ferguson, and for an introduction, I'll turn it over to Dr. Pasquale.
Hi, good morning. On behalf of the Physical Medicine Rehabilitation section, it is with great pleasure that I welcome Dr. Adam Ferguson today as our featured lecturer for today's grand rounds. Dr. Ferguson is a professor in the Department of Neurological Surgery at the Weill Institute for Neurosciences here at UCSF, a principal investigator at the VA Medical Center, and the director of science in the Brain and Spinal Injury Center at Zuckerberg San Francisco General. Dr. Ferguson received his MS and PhD in behavioral neuroscience from Texas A&M University. He has since received numerous awards, including a postdoctoral NIH National Service Award and an NIH Early Stage Investigator Award. His body of work has expanded to several hundred papers and conference proceedings focusing on the area of expertise for which he is internationally known, and that is in research in neuroscience and the role of big data analytics for both scientific discovery and also for decision support in healthcare. I know Dr. Ferguson best for his work in spinal cord injury research, where we work together in the TRACK SCI research group at ZSFG, for which he is a principal investigator and co-director of the International Open Data Commons for Spinal Cord Injury. Many of you know him as well for his work with the Backpack Initiative here as well. And thank you, Dr. Ferguson, for spending time with us today.
Thank you, guys. It's a great pleasure to be here, and thanks for having me. I titled this Leveraging Data Science for Rehabilitation Research, and then Dr. Pasquale pointed out it doesn't have to be limited to that, so I expanded the scope a little bit. I do have some disclosures. I have a number of grants that I'm either PI or co-I on. I do some data science consulting, and I have in-kind support from an industry collaborator. I won't be talking about that in any detail today. The premise of my talk is that CNS injury is complex. Central nervous system here is, of course, both the brain and the spinal cord. In the human brain, we have trillions of synapses. And then with traumatic brain injury, you superimpose on these trillions of synapses random heterogeneous injuries, and you're going to generate enormous complexity. It's going to be hard to predict outcome. And yet, clinically, the field of neurosurgery limits itself to 3 categories for describing TBI. So trillions of synapses, and then you have mild, moderate, and severe. I'm going to be arguing today that that is insufficient for characterizing the disease with any precision. Spinal cord injury is not much better. We have billions of synapses in the human spinal cord. It can do all kinds of processing. And then the major outcome that people typically use is ASIA grade. And, I think Dr. Pasquale just pointed this out. It's A through E, where A is worst, E is best. So 5 points. And again, I'm going to argue that is insufficient for predicting outcome and understanding how spinal cord injury manifests in the real world. In the preclinical literature, we sort of have the opposite problem. People focus on small details of the biology. And so the literature is filled with gazillions of tiny measures of biofunction. And you'll see these papers that have these multi-panel figures where things go up, other things go down. And in the discussion section, they try to sort of synthesize this. And I'm going to argue today that this level of detail is also insufficient for describing central nervous system injury. To do this well, we really need to start with granular data and then get serious about the way we organize our data so that we can deploy machine learning tools and advanced analytics to help us understand the nature of this biology and how it predicts outcome. So this really comes down to this poor soul here. So if you're going to do big data research, someone has to manage the data. And at the current time, this someone is a resident or a postdoc or a graduate student who, instead of pipetting, sits in front of spreadsheets and organizes data. So I want these people to be celebrated as like the great heroes of this talk today. So this way of thinking kind of recasts CNS injury, not so much as a problem of biology or biomedicine, but really as a problem of big data. And it's a specific type of big data problem. This term big data was coined, in Silicon Valley in the early aughts by Gartner Consulting, where they're trying to describe the the current problems with data management. And they basically said there are these canonical 3 V's that describe big data. And traditional databases aren't very good at handling these. First was the problem of large data volume. The second was the problem of high data velocity. So this could be small little bits of information flying around very quickly, like in the ICU or in the Twitterverse. And then there was this third V of big data, variety. This idea that if you have heterogeneous variables and a great many of them, it's hard to manage that data and use it meaningfully. I think in biomedicine at large, and in CNS injury in particular, our problem is not so much volume and velocity. Our problem is that we have a very wide variety of data. Outcome from traumatic brain injury, spinal cord injury, stroke is not one thing. It is a great many different things. There are many variables that characterize the disease. And this is sort of a smaller form of a larger problem in biomedicine that we have historically not been a sophisticated data management field. If you plot the number of data sets that are reported in the literature against data size, you can see that there's actually relatively little organized big data in the field of biomedicine. Most of the data that's collected falls in what they call the long tail of the distribution, where you have a great many data sets of modest size with wild heterogeneity. And at a certain point, you reach the literature limit, and beyond this, you have what they call the long tail of dark data. This is data that is collected, it's very heterogeneous, might be small little bits of research data, and then you put it in a file drawer, and you never publish on it, you never use it, it didn't support your hypotheses, you didn't know what to do with it. Based on meta-analysis, we now know that this dark data comprises 65 to 85% of the data that's collected in biomedical research, which is actually kind of problematic if you consider how much money we spend on biomedical research, which is about $300 billion a year worldwide. And so if you take this number and you multiply it by 0.65 or 0.85, you end up with this crazy conclusion that our biomedical research enterprise generates about $200 billion worth of research products per year that go into a file drawer and are not disseminated in any way. It's like the vast majority of our information is lost or dark. It's also statistically problematic, so it's costly, but statistically problematic because it means that the published literature contains something like 15% of the data that's reported. You could ask yourself, what is it that's so special about that 15% of the data that makes it into scientific papers? And there are some biostatisticians who have made the claim that those 15% are those 15% that are most supportive of me being right about my hypothesis. And so we, by doing hypothesis-driven research, have a tendency to sort of say, I don't understand this data, it doesn't make sense to me, so I'm tossing it out. And then you run the experiment again, collect more data, and you go, oh, this time it looks like I'm right, I'm going to write this up. So the consequence of that is that the published literature is extremely biased. It does not reflect all of the data that was collected. And that's led some biostatisticians to claim that most published research findings are false, meaning they would not, as a statistical phenomenon, be expected to be randomly reproducible. And this has actually been shown through meta-analysis. And when people have tried to replicate high-impact studies, they find most of the time they cannot. And the popular media picked up on this. There are some articles in The Economist, New York Times, and others that were saying, science is not as self-correcting as we think it should be. And this led the NIH to come up with this new need for transparent reporting to try to improve preclinical research. And then, of course, the NIH Reproducibility Initiative, which those of us that write grants are very familiar with this now. So the idea is that we need to improve the degree to which the literature reflects the data that is collected. And I don't think this is actually anyone's fault. It's really just an artifact of what we do as scientists or biomedical researchers. We spend all our time gathering these beautiful data using airtight protocols. And then, to become a full professor at UCSF, I had to publish 190 papers to get there. I had to somehow take these thousands of data sets and winnow them down to, like a less than 10-page high impact paper, hopefully in JAMA. And in doing that, there's going to be loss of information and potential for selective reporting. So the problem with this whole publication bias issue is that we do have a data dissemination technology in science. It's a 17th century technology. It's the scientific paper. So this is the first scientific paper that was written in English in 1667. It had no data in it. It was a narrative about data. And in essence, that is what we've been doing ever since. Scientific literature is narratives about data. You might have statistical summaries of data. You have conclusions about data, but the data themselves are not in the paper. So that's an important point. And as a consequence, you don't know what all the data was that was collected. So the NIH and several funders have decided that they're going to change this system by now demanding that people make their raw data publicly available. So you get credit for the data you collect rather than the story that you tell. And the idea here is that data sets themselves, scientific data, should be made findable, accessible, interoperable, and reusable, FAIR. If you haven't heard this acronym, you will as soon as you put in your next grant, because they've been thinking about this for a while. The National Academies of Sciences had a bunch of work groups that I was fortunate to be involved with, where they looked at what it would take to fully support data collection and data publication as a major work product in biomedicine. They talked about how you could do this in the context of neuroscience in particular, and then what it would take to do, culture change to ensure that we're sharing our data. And then there's this new policy that NIH released that goes live in a month and a half, basically, 2 months if you squint. So January 25th, 2023, every person who puts in an NIH grant is going to be required to have a data management plan that articulates how they're going to make their data fair and open and accessible and citable at the end of the grant or upon their paper publication. And if they don't, they won't get their next grant. So they're trying to really force this cultural change. For our part in spinal cord injury and TBI, we're kind of in a good position for this because my first R1 actually at UCSF was to go gather as much raw data as I could in the preclinical literature. And to my great surprise, the spinal cord injury field was like, yeah, man, you can have all the data. Like, here, have it. I'm done with it. I published my paper. I've already gotten my piece here. So we're able to sort of quietly assemble this large repository, 4,000 rats and mice and monkeys with spinal cord injuries and human medical records that are de-identified from 13 centers. And that was the basis for this thing that was called the Vision SCI repository. This has since been expanded and picked up by all of the nonprofits in the spinal cord injury community. They're pooling their resources to help the community build this open data commons for spinal cord injury. I'll talk more about this in a bit. And based on the success of this effort, the NIH and the VA asked me to put in a grant to do the same for TBI. So now we have these two very large infrastructures that are designed to basically manage data and publish data, almost like a journal. We've had partnerships form with a number of federal funding projects that are using these two infrastructures as their core sort of data management, data dissemination technology. And we have these partnerships with very large clinical studies that are running out of UCSF, Track TBI and Track SCI. These are clinical observational studies that are designed to sort of characterize what it is to have a traumatic brain injury with all the data that you can get, or an acute spinal cord injury with all the data you can get. And these groups are minting de-identified versions of their data sets that we put up in the open data commons for spinal cord injury and TBI. So early on, when we were trying to get people to share their data, we had a meeting at NIH where we asked the spinal cord injury community, under what conditions would you be willing to share your data? This is in 2016. And what we heard from people is that PIs did not want to just upload their data to a publicly available cloud like Facebook or something. They wanted to have control over the data management in kind of a cloud space that was secure. And then they wanted to be able to steward this data into increasingly public spaces. So we established this sort of private lab space that's only available to the PI and the lab members that the PI lets in. And then the PI has the ability to push data into this sort of preprint space that's available to all verified labs. And indeed, they can actually request that they get a digital object identifier and make the data truly published. And this kicks off an editorial review process, kind of like a paper. And when it's done, they have a persistent identifier for their data set that is citable. And because it's a CC BY license, when people reuse the data, they need to cite the authors or it's copyright infringement. So there's a way to enforce this as a new metric for academic productivity. So this is kind of what the portal looks like for spinal cord injury. At the current time, we have 98 labs, 191 data sets. This is over 10,000 individuals with spinal cord injury, rats, mice, and humans. We have 35 data sets that have been publicly minted into DOIs where people have chosen to publish them. This is almost 4,000 individuals. So you can now, if you want to do data mining, go to this website, download 4,000 individuals with spinal cord injury data, and do your thing. You need to cite all of those 35 papers or data sets when you reuse the data. In TBI, we're a little less far along, but it's coming. And I just want to sort of show you guys what this looks like. If you explore public data, it'll drop you into this page. It's the landing page for this data set. This DOI is resolvable anywhere in the world. If you click if you search this in Google, it will come to this page. You get the data set file. So that's the just rectangular data matrix. And then the data dictionary that describes every variable. And those two describe the citable unit. So you see here, there's a data set citation. So this is the official index worldwide by libraries everywhere citation for this data set. In addition, because it's this Creative Commons attribution license, people must cite this data set when they reuse the data. And if they don't, that's like plagiarism, basically. We've had a lot of uptake in the spinal cord injury community. Every continent except Antarctica, we get regular visits. In the US, it's been pretty well taken up. So it's coming along. So anyway, all of that is supported by federal funds and is nonprofit. And if you want to share data of that form, TBI or SCI, I encourage you to go check that out. Sign up, get a username. But what I want to do for the rest of my talk is talk about what one can do once you have big data that's publicly available of this sort. And the basic idea in our lab is that we want to do machine intelligence-based data fusion. So you have all these multidimensional variables that have been collected. If you take the case of sort of grasping function in the context of neurorehabilitation, you could sort of quantify the quality of grasping using an ordinal clinical scale that's maybe like a 1 to 5 scale. Or you could do some very sophisticated neurophysiology to quantify that. And our position is that we don't think one of these is better than the other. We just consider them two different pieces of information that describe the individual. The individual has a neurophysiology score and a grasping function score. And this individual, by the way, could be a rat, monkey, mouse, human, doesn't matter to us from an analytic perspective. What we do care about is how this individual stacks up against all other individuals. So you can do this in like a 2-variable space or a bivariate space, or you can do it in a multivariate space. You can even do it across different types of data. So you could have the full set of all functional changes, the full set of all surrogate biomarkers, and the full set of tissue changes. If you can track the same individual across these different data layers in multiple dimensions, this allows you to then cluster individuals based on their similarity. And you get a sense of what it means to be a member of a group in this sort of multidimensional space of the disease you're studying. So when you're doing this with patient data, this becomes sort of a tool for precision medicine. The idea is you have all the variants that you're measuring across all the variables, and you can ask yourself, to what extent do these patient groups overlap or are they separable? So then that becomes a very powerful tool for precision health because you can say, I want to take individuals that are over here in this multidimensional cloud and try to nudge them so that they look more like the ones here. So you're not limiting your therapeutic to one variable now, one arbitrarily chosen primary outcome, but all data simultaneously. The same thing can actually be done for translation across species groups, and it can be done across laboratories for reproducibility. So I want to sort of illustrate how you actually do this. I'm going to start with an example of reproducibility and data reuse. So this is from a project at the San Francisco VA that was run by Steve Massa, Ray Swanson, and Jailing Liu, where they had like 3 different labs running this large sort of program project grant in rats, where they're looking at rehab, anti-inflammatory therapies, and pro-plasticity therapies in the nervous system. And they're trying to improve recovery of function after traumatic brain injury. So as is sort of typical for these types of studies, they had many different outcomes. They had like the histopathology, they had a bunch of motor behavior, learning and memory tests. This ended up being like 30 outcome variables taken over time, which ends up being potentially up to 6,000 t-tests that you would look at if you were going to try to find a significant effect. So then the question is, what would one do with this kind of data? And what people have done historically in the field is they'll say, I like the way this graph looks, so that's going to make it into the paper because that really supports my hypothesis. And what Jenny Hafley and our team realized is that since she knows how to code, she could just do every possible statistical test. So she did that. And what we have here is a plot of the p-value for each test, asking whether there's an effective drug or rehab, and then the number of tests that yielded that p-value. The first thing I would note is that most of the effects were not significant. There were a few that were really itsy bitsy. And another interesting point is that one right there is 0.05, which was just as probable as a p-value of 1. So the idea here is that this p is less than 0.05 thing that you see in the literature isn't necessarily a measure of reproducibility. Nonlinear principal component machine learning approach, where she asked, how does tissue sparing general health measures and functional behavioral measures overlap? And finds the variance that is shared across these. And then from this, she can come to an understanding of what the variance cluster represents by looking at the correlation between individual variables in this cluster. And what she sees is that brain lesion is related to, learning and memory and attention, motor function, sort of makes sense. But what she was able to do is from those 6,000 data points for t-test, now she has one variable, this principal component 1, that accounts for about 24% of the variance in the data set. She could then project each animal into this syndrome space that's defined by this PC1. She sees that TBIs are here and shams are over here. So now she knows which way a therapeutic effect should occur. It should go up on PC1. So then she can flip the coin once and ask in a hypothesis-driven manner, is there an effect of a neurotrophic drug, rehabilitation, anti-inflammatory drugs? And with that single coin flip, she's characterizing many hundreds of variables at once, was able to show that there's this neurotrophic drug that had a modest effect that was significant. In addition, rehab, PT, had a pretty large effect on recovery. And unfortunately, the PT and this neurotrophic drug actually were competing for plasticity and they canceled each other out. So if you did rehab plus this drug, that was much worse than doing rehab alone or the drug alone. So that was sort of an interesting finding that had been in the literature theoretically, but was not discernible with the data that this team had. So this is a way to test reproducibility. And the thing that was interesting to me is that this is kind of a fun paper that we wrote. It was kind of esoteric and mathy, as you guys are seeing. And to our great surprise, Nature Medicine got a hold of it and said, this is like super newsworthy because we think that this is an example of how one could find drugs and doses using artificial intelligence by taking advantage of all the data. So they gave us a megaphone for that, kind of little fun romp that we did. So we've now moved on from that initial work and begun building tools that will allow others to do the types of analyses that Jenny did. Abel Torres Espin and our team, who also works with Backpack and Reach, has developed this R package that came out in eLife that allows you to, in an automated fashion, do a bunch of advanced sort of machine learning stuff. And it's free, open, and using this tool, we're starting to now apply this type of methodology all over the place. So one example is to look at kinematic function after spinal cord injury. So again, kinematics can have hundreds of variables that you're measuring, and usually our ends aren't thousands of individuals. So how do you manage that variety of data? So we'll do these principal component analyses that will tell you the chunk of variability that is very systematic. Then you can ask, what's the effect of patients versus controls? What we see in spinal cord injury is that there's a dispersion of the variability in the kinematic patterns in patients relative to controls. And as an ensemble, you can distinguish the kinematics in patients from controls using this AI. So this is an application for precision health. And I guess a more close to home example of this same application is this work by Anastasia Keller working with Jeannie Bailey and Rob Matthews in rehab and orthopedic surgery, where they were applying these same types of approaches in the context of low back pain. So the idea is there were these three-dimensional kinematic markers of movement in the sit to stand test. And you could measure the points in space over time. And then it becomes very hard to sort of look at this and understand precisely what the differences are. So rather than focus on any one parameter, Anastasia and Rob took the whole set of all parameters, again, did this nonlinear principal component thing. And they were able to distill the kinematics down to this cluster that was, 37% of the variability. It was comprised of all these sort of specific measures, and they can name it as vertical rise in standing posture. So this is a mathematical quantification of this standing posture. So now you have this one variable, and you can ask, is there an effect of low back pain on this? And what she shows is that the PC1 was significantly able to distinguish low back pain from control in sort of a linear fashion, a stepwise fashion. So non-specific low back pain was, nudged over on PC1, and then spinal deformity, a more extreme version, was nudged even further. In addition, she could see that the VAS back pain measure correlated with this PC1, and the ODI measure also correlated significantly with PC1. So the idea is by taking machine learning on visualization of movement, she could predict patients in their clinical grouping just from their movement. In addition, there was this separate principal component, an orthogonal axis that sort of comprised the leaning strategy and biomechanical consequences of that leaning strategy for standing up. And again, she could see that there was this discrimination on PC2. So altogether, we now have these two axes from many hundreds, PC1 and PC2, that characterize over 50% of the variance in kinematic movement. And they seem to be very sensitive to low back pain groups. And so in other words, low back pain goes this way along this diagonal, and you would expect a therapy to then nudge individuals back in this direction. So that would be sort of a precision medicine application of this math. And so they were able to sort of quantify these sort of qualitative features in a very rigorous way here. And then Anastasia, for those that were at the Journal Club, we covered one of her prior works. She's really into this idea that neuromuscular pain and these kinds of mechanisms are targets for spinal modulation, potentially. So you could actually apply spinal neuromodulation and potentially improve, she hopes, posture and maybe then low back pain. And there's a good reason to think about this. There have been a number of papers where people have looked at non-invasive reactivation of movement with trunk stability, and then of course the paper that we talked about today. And I would note that in this first paper, we did these nonlinear principal component analyses to characterize the therapeutic effects that these other papers then built on. Okay, so So I want to now pivot a little bit to this idea of bench to bedside translation and the way that you can use machine learning to do that. So this is a kind of interesting story because it began um, in a pub in Potrero Hill, where a young guy came up to me and said, "Hey, like, what do you do?" And we got into this thing where, "Oh, I'm a professor, at UCSF. I'm just starting my career." And he's like, "Cool. I'm a bioinformatician looking for jobs. I've been in industry and I kind of want to come back to academia." So he came and interviewed in our lab to take a job as a computational person. And then sort of said, "I'm not going to work with you guys. I found this really great job at a startup down in Palo Alto, so I'm going to do that instead." Okay, understand. Have fun. Have a good life. And then like 3 weeks later, he wrote me and was like, "I just can't stop thinking about how important spinal cord injury and traumatic brain injury are as problems. And I worked for this company that's built this machine intelligence tool that undoubtedly is going to be used for like banking or something, but it might be fun to look at biomedical applications." So I went down, set in the duplex, opened the fridge, elbowed the computing cluster as like a stereotype of a startup. And it turns out this thing was this project that had been DARPA funded and was a top secret project for understanding how individuals are similar. You can imagine how that might be used by the military. And they had spun out this SBIR company where they're trying to come up with civilian applications for this tool, and it's known as topological data analysis. So we decided we'd form a partnership. We got a grant together, and explore this type of advanced machine learning to problems like traumatic brain injury and spinal cord injury. And the way this stuff works, in essence, is it basically looks for these points of overlap among clusters, and it represents those points of overlap as edges. And then individuals that are indiscernible are shown as nodes. And then it iterates through gazillions of different variables and comes up with sort of a consensus version of what it means to be a member of a group. To sort of test out this technology, we chose really the most old school 20th century data set that we could get our hands on. There's this multi-center animal spinal cord injury study that was run in the mid-90s that was the source for this idea that people should give steroids for spinal cord injury, which has now been abandoned as an approach. Part of the reason that it was abandoned is this study didn't actually show any results. They collected all this data, went into these file drawers, the hypotheses were not supported, and it vaporized, like was never reported. So my postdoc, Jessica Nielsen, and a couple of her technicians digitized all this stuff and annotated it and gathered the full set of records. And then once that was done, we could then throw it up into the cloud and do this advanced machine learning on this 20th century data. The first thing that we saw is that there's this network map that represents spinal cord injury, rats. 390 rats are shown here. And you could just paint this network by variables of interest. So Jessica navigated this and saw that locomotor recovery was different in these groups. These did very poorly. These other animals did very well. So she could ask, why are these different? The most obvious one is they probably had different injuries because, these are biomechanically controlled injuries. So we looked at that, and that was not the case. In fact, they were indiscernible in their injuries. So then when I see this, I think we might have just found a drug therapy effect that was hidden in Mascus, and we're going to be heroes. And so I asked her to do a supervised machine learning thing where you ask a machine to tell you why these are different of all the many thousands of variables at our disposal. And to my dismay, I was wrong. It had nothing to do with drugs. And actually, this is a principle. Machine learning does not care about what we think. It was telling us that the thing that mattered most was blood pressure. And in particular, animals that had high blood pressure had poorer outcome. So that's a clinically actionable thing. You could imagine that if someone's having a spine stabilization surgery and you could monitor the blood pressure to try to avoid high blood pressure, that might yield long-term locomotor recovery like you saw in the rats. Problem is, when I say this to clinicians, they're like, machine learning is a black box. I'm not going to change my clinical practice based on you and your sort of zany math that was discovered in a bar, right? So to try to build a case for reproducibility. Red Bull, Wings for Life, gave us a grant to go get all the rest of the Damascus data, which had been in a storage unit in New Jersey for 20 years. And so we went there. Jessica had her Explorer hat on. We recovered data and digitized all this. We're able to end up with an N of 1,400 rats, which now have data sets that are up in the Open Data Commons. And this whole story yielded this finding that there is indeed a blood pressure relationship to long-term locomotor recovery in rats, and it's nonlinear. So there's this point in sort of the Goldilocks zone where blood pressure management very tightly can yield better outcome in rats. So this is a successful replication of Jessica's study. And then, of course, there's this fact that rats are not human. So what about translation to clinical care? Well, as Lisa mentioned, I'm very lucky to be part of the TRACK SCI study at the Zuckerberg San Francisco General Hospital. So this is a clinical observational study, has every service that sees the spinal cord injury all working together to build data sets. From this, we're able to curate 98 surgical records from the general and add to this 33 surgical records from Santa Clara Valley Medical Center, pooling this to a cohort of 118 patients. And then applying this topological data approach, first we could actually look at the blood pressure. So each row here is an individual subject and 118 rows. And the color is their blood pressure in a 5-minute epoch of time. And here are the heart rates. So we have this sort of physiome of function after spinal cord injury in the spine stabilization procedure. So from this, we could then yield a distance matrix across humans. We could project every human into this multidimensional manifold and then cluster humans based on their similarity. And so, we have these groups here. The patients are nodes. When they're similar, they have these edges. So you can distill this down to these topological clusters. There are 11 of these sort of phenotypes of the physiology after spinal cord injury, or physiome of spinal cord injury. We could then rank these clusters by their map. And what we saw is that individuals that had maps that happened to fall within this middle range had much better probability of long-term locomotor improvement. So if blood pressure was either too low or too high, these patients had much worse outcome, which is, you could argue, the end of this story that started in Potrero Hill in a pub. We have a successful replication and now translation to humans. And so this is now modifying the way that the neurosurgeons are doing care at the General Hospital. And prospectively, we're starting to come up with expert augmented machine learning approaches where we can commingle machine learning with clinical input to try to improve outcomes in spinal cord injury. And so I think this is sort of a step towards the future. And I'll just sort of point out that some of this stuff, in addition to being just fun math, also may matter. We had one of the individuals in our study happened to be enrolled when the General Hospital was now paying attention to blood pressure and interstitial pressure management in the spinal cord. And this guy came in as a high cervical injury, complete quadriplegic after surfing accidents, and is now able to walk, drive, swim. And so maybe we have something that we've discovered here by doing machine learning, and it may actually have impact in the end. So I'll stop there. This is where I work. These are the people I collaborate with, and thanks. I'll take questions.
Thank you, Dr. Ferguson. That's fascinating work and very impressive. Thank you for coming to join us today. And any questions from our audience? I'll start with Dr. Lotz.
Thanks, Adam. And we're super fortunate to have you collaborating with us. So appreciate that. You're a great example of a great collaborator. So everybody pay attention. So my question is, you mentioned you're looking at kind of existing measures and you're looking for these patterns. Has it been, is there approach then to say, what are the better measures that might be used in the future, kind of going forward, or developing new diagnostic tools?
Yes, one application of these approaches are to take the hundreds of measures and then distill it down to these principal components. And then you can cherry pick the variables that are most highly predictive of those principal components. So it's one way to kind of filter the data. There are more elegant ways, which are like these penalized regression models and support vector machines. There's Patricia, she's on there somewhere, that do the same kind of approach. They say, I want to come up with like 1 or 2 variables that are most predictive, most robust. So these types of approaches can be used in that way as well.
Thank you.
I had a question. This is Tamara Alliston. Great talk, Dr. Ferguson. Thank you. My question is similar to Jeff's in the sense of, we have the choice in the lab to decide what outcomes are we going to collect on any given thing. And, in our case, it's more about bone or cartilage outcomes. We can choose things like RNA-seq or mass spec, where we get unbiased data, or we can choose more targeted things where we're looking at one specific gene and one specific set of conditions. And how based on your experience in these analyses, what are the most powerful sets of data to help us guide the important principal components?
Yeah, I'm a fan of trying to begin with as much data as possible. And I think one of the dangers of hypothesis-driven research, where you myopically study the thing you care about, is that you may be creating a sort of selection bias, where you say, when my hypothesis is supported, then this is real. When it is not supported, I think it's not real, and that can introduce some bias. So I'm a big fan of gathering all data, especially if you don't know what to measure, and then doing these data-driven approaches to help filter it down to a small number of variables.
Hi, thank you so much. I'm Rosie. I'm one of the tumor surgeons here, so I know nothing about this topic, but I found that such a fascinating talk. I also want to say thank you for putting everyone's photos up there. I thought that was really nice to see who's behind the work, and so kudos for the talk. I do have a question coming from an ignorant place, and maybe you and the spine surgeons can help with this. We often discuss cases in our M&M conference where during an elective spine surgery, a patient loses motors. And then, because there's constant neuromonitoring, and then, they raise the blood pressure, do something different with the decompression, and then move on. Would this be a scenario where your technique might work, where we have all of the data of the maps throughout the case, we have all the neuromonitoring, we have an operative report? Could we take all the past 10 years of our data and maybe find things associated with that?
Yes, absolutely. Yeah, it would be a. This is why my postdocs hate me. It's really easy for us to do this if we have a collaboration, because I have a lot of very talented young people I work with.
Yeah, terrific follow-up to the Journal Club, and the power that you're showing in reanalyzing some of these large data sets is important. We talked in the first session about, well, the tool butte and the idea there would be a travesty if we didn't have some information that came out of the trillions of data points we have. Yet what data is missing? And specifically in your work, say with spinal cord injury, what's missing with regard to intrathecal pressures, with regard to other omics, right? Our genomics, our transcriptomics, our radiomics. What's missing that's not in a data set now?
Yeah, so we do have some transcriptomics that we're collecting from the blood. But spinal cord injury, to my knowledge, doesn't have GWASs that have been run. So maybe behind some other fields in this way. The radiomics is a challenge, of course, because spinal cords are relatively small and you need certain MRI sequences to see them. So there's quite a bit of missing data that I think we could, if we could gather more information on spinal cord injury in a unified way, we'll be better positioned to hopefully have other discoveries like this, maybe more important discoveries than this.
I think that's where there's got to be a real commitment of some effort, is to actually increase the baseline of the data, because there are variables in there that aren't included. Have you had a chance to access the UC Rex system at all in terms of research exchange? And is that becoming something that's useful yet? Because it hasn't been so far.
Yeah, I think it has a lot of potential. Some of the folks in our team are really into real world data, and they have sophistication and seem to be able to get things out of UC Rex. The problem is brokering access to these highly granular data across the UCs, I think, is still a moving target. how do we, what are the consent issues and things of this sort.
Hi, great talk, Stephen. Being I dabble a little bit in the space, and it's really fascinating to hear. I have you did lose me with the P05 not being effective, so I'm going to go back to this again. But why? Yeah, I'm sure it's a simple thing that I just think might get. But the other question is, looking at the way you're collecting data and storing it, and the NIH is requiring it to be accessible to everybody. That's a question mark I have, because how do you actually, well, who's going to be paying for the storage, and how long is it be stored for, and how is it going to be accessible, and how do you, that whole piece about even who gets access to the data, is it accessible for commercial purposes? I mean, how you guys are thinking about that, I wouldn't mind. I would love to go over this again if anybody else has wanted to, but the other question is about the structurally how to continue to make that data accessible over time.
Yeah, it's a challenge. The way to think about this is a library sciences problem. And these National Academies reports, if you're really needing to sleep, this thing's, each one of these like 300 pages where they go at length into this, especially in this one. They talk about the different sort of phases of the life cycle and how much it'll cost. NIH is aware it's going to cost something. The way it's going to be enforced is not through peer review, but is programmatically. So this will be like a box checking thing potentially, where if you don't adhere to making your data fully accessible to the extent that you can, not human identified data, but all the rest of your data, and they're going to enforce it. How are you going to pay for it? Well, they're going to allow you to put it into your grant in the same way that you might have publication fees in your grant. But of course, like every new policy, this is going to be a long slog and a rollout, and we all learn together how to do this. And it's hard to argue that in the end it will be bad, but I can say at first it's going to be pretty rough for everybody, I think. Yeah, so they argue that it should be accessible with no restrictions in this policy. And again, if you want to go to sleep, you can read the whole NIH policy. It's pretty extensive. The idea is there should be no restrictions. The taxpayer pays for these data collections, and if the taxpayer wants to start a business using those data, that should be allowed. The CC BY license demands that those reusers cite the original authors in the same way that the patent system requires citation. Yeah.
Hey, my name's Stephen Garcia. I'm one of the Orthopaedic residents. Thanks for the talk. My question is, when you integrate a lot of these heterogeneous data sets, is there a way to tell what data sets may be of poor quality, or how does that play into kind of these mega analyses? I do a lot of like transcriptomics, so I'm really curious about that question.
Yeah, the QA/QC question. It's also frequently phrased as garbage in, garbage out. The truth is, once you have enough data, these things start to normalize. You can say this particular group or data set seems to be an outlier in this multidimensional space. That can be one sort of key. And there can be opportunities for sort of, detective work to try to figure out why groups get different results. Right now, what people do is they publish different papers and they yell at each other in rooms, right? There's no way to really adjudicate that. So this, I think, will help having access to the data. Exactly, yes.
Hey, my name is Kaz. I'm one of the residents. I think if I understood correctly, basically one of your points is that hypothesis-driven science is basically leading to a lot of the data being ignored, as you were mentioning. What do you propose as move away from hypothesis-driven data in the era of big data? And like, how can we start reforming how we're asking questions, or should we not even ask questions and have an algorithm think of which questions we should ask for? But where do we move from getting away from all that dark data?
Yeah. So I still think hypothesis testing is extremely powerful. And we did that here. We've discovered something using machine learning. It pointed to a problem we then chased down with an inferential hypothesis driven sort of narrow scope. The danger is when you don't know what to measure. And that's where these data-driven approaches are really powerful. The way I think about it is the scientific method that we all learned in elementary school was observation, form a hypothesis, test a hypothesis. And when you have multidimensional data, it's that observation piece that's super hard, right? Because there's thousands of observations you can do. We're not designed to look across thousands of columns of data. As you know, our human brain is not designed for that. So this is observation that I'm talking about. And then hopefully that will lead you to a hypothesis that you will rigorously test.
Great presentation. Obviously, from the questions, you're really making people think. And I wanted to go back to this notion of the data sets and the missing data, because as clinicians collecting data, we stress over something's missing. You don't have the same set of data on every patient you've tried to follow. Then thus maybe rendering your data not useful. I just was interested in your perspective as a data scientist on that stress, because it sounds like you think, well, that data is probably still meaningful if we combine it with something else. Is that kind of how you think about it?
That is how I think about it. I think there's no such thing as bad data, you know. And I think that if you have more data, it is possible that data could be manipulated, but if you have more variables, people are not going to be able to manipulate all 10,000 variables on a patient, you know. So I think having direct access to the data is key. When data are missing, I guess we talked about this in yours. Yeah, that's right. There are ways to sort of deal with missing data, and that's a whole branch of statistics. I won't get into exactly, but there's a book by Little and Rubin called Statistical Analysis of Missing Values, which one of my neurosurgical colleagues saw me reading and said, "Boy, that looks like a page turner, you nerd." You know, but it's one of the most highly cited works in all of academia because it's so useful now for this exact reason.
So you may have noticed most orthopedic literature still uses bar graphs, and if we get complicated, we use lines. Should we be transitioning our research to a bunch of graphs that we can't understand? Is that a better way to transition, or is there still value to do these relatively straightforward experiments and look at relatively concrete values?
I mean, I do think that there's value in science and doing the science you know how to do. I also think there's value in your raw data. I guess that's my claim here. You can't expect orthopedic surgeons who are busy in the OR and saving lives and improving lives to take time out of their schedule to learn how to code. That's just not a reasonable thing. You do you, and then we'll also have your data if you don't mind.
All right, so I think we'll wrap up there, but Dr. Ferguson, thank you so much. This was great and definitely appreciate it.